Hello everyone. In this video we are going through the theory of the practical technique of serial dilution of a bacterial culture in order to determine the cell density in the original bacterial culture. So remember, in any practical investigation, there's the factor that you are going to change, otherwise known as the independent variable. You are going to keep certain other factors the same, constant, unchanged, because they are your control variables, okay? So independent variable, control variable, and these two in combination allow you to make some valid measurements that will allow you to make a conclusion about what effect changing this factor had. Okay, so the first thing is what you're changing, 2 is what you're keeping the same, and 3 is what you measure. Okay, and this is otherwise known as the dependent variable. Okay, now any investigation at any level ultimately consists of these things. Yes, there are other considerations around safety, around reliability and accuracy, around analyzing your data, even ethical considerations. But this forms the core of any scientific investigation. And like a lot of techniques that you will learn about in biology, the thing that you might spend most of your time understanding and revising and making notes on is or are going to be techniques of measurement. Okay, so how to measure the rate of an enzymic reaction, how to measure the biodiversity of a particular habitat, how to measure the rate of diffusion of a pigment out of bits of beetroot, things like that. And bacterial serial dilution is another one of those techniques. And so it doesn't stand on its own. It's a technique of measuring something, but by itself, it's not a whole experiment. Okay? So, where bacterial serial dilution and culturing comes in is the measurement of bacterial cell density, or in other words, you could call it bacterial. It's a way of measuring bacterial growth. Okay? So if you are asked, or if a question asks for you to design an investigation that requires the measurement of bacterial growth, this is where the technique of bacterial serial dilution comes into it. Okay, so maybe you're investigating the effect of temperature on bacterial growth, or the effect of pH on bacterial growth, or the effect of some antibiotics, though there are other experiments that also look at that. But if you're looking at how something affects bacterial growth, you're going to do these things, you're going to change that factor, you're going to keep other things the same, and then at the end, you're going to do what we are talking about in this video. Okay? You are going to measure the bacterial cell growth. So how do we do that? What is the technique of measuring bacterial cell number? Okay, so essentially we are talking about the serial dilution. Now, serial dilution can be applied in a number of different contexts, chemical, biochemical, and also microbiological, which is what we are doing today. Now, so as the theory goes, with the serial dilution of bacteria, it all really starts with a vial of bacteria with an unknown density. And your technique should allow you to measure, or estimate at least, the number of living bacterial cells in this original culture. Okay? So how I would explain this is essentially on a very most basic level, if you wanted to find out the number of cells that are in this solution, what you'd basically do is take a known volume of that bacterial culture, okay? So let's say we took 100 microliters of this solution, or of this suspension, put it on an agar plate, we incubated it for 24 hours, incubate 24 hours, and then we get some colonies. So every single cell from here that was living got spread on the plate, and every single cell multiplied multiplied over those many hours. One cell became 2, became 4, became 8, became 16, became 32, 64, until after 24 hours you could be in the order of millions of cells that have formed now something that's visible to us as a colony of identical cells. So if that happened to each of the cells that we put onto the plate, we get a certain number of colonies that we can count. So we incubate 24 hours, we count the number of colonies, okay? And because we know that we put 100 microliters of the original culture onto the agar plate, then we know that in the original 100 microliters, we had 1, 2, 3, 4, 5, 6 cells. We had 6 cells that became eventually 6 colonies after 24 hours. And we had 6 cells in 100 microliters, so if we had 6 viable cells or colony-forming units, 6 cells or colony-forming units in 100 microliters, then we have 60 colony-forming units in 1 ml. Okay, so this then gives us an estimate of how many bacteria, or what was the density of the bacteria in the original culture. Okay, that's in the most simple scenario. The thing is, bacterial cultures are often very dense, and the likelihood is that if we did this with a normal bacterial culture, we wouldn't get 6 colonies, we would get so many bacterial cells all bunched up together that they would form one single monolayer of cells on the plate, covering everything, giving us nothing from which to be able to calculate the original density of the cells. So to circumvent this problem, what we do is we do the serial dilution method. What that means is we don't just take 100 microliters and plate it directly onto agar. Because we don't know how dense this culture is, we are going to do a number of dilutions where each culture is more dilute than the previous one, or each suspension is more dilute than the previous one, giving us a range of bacterial suspensions of known dilution, right? Okay. So we are not going to put it directly onto an agar plate. What we are instead going to do is set up a series of tubes, right? And we're going to add 100 microliters. So we're going to add to all of these tubes to begin with, we are going to add 900 microliters. 900 microliters of nutrient broth. Okay, so all of these tubes will have 900 microliters of broth, and we are going to add into the first tube, so we'll call this tube A, B, C, D, and E. Into the first tube we'll add 100 microliters of the unknown bacterial suspension, which means that this first tube has a dilution factor of 1 in 10. Okay, fact, just put that here. Okay, so that's got a dilution of 1 in 10. We are going to invert that tube a number of times. we are going to then transfer 100 microliters from there, from tube A into tube B. But by this time, because we've diluted that again, 1 in 10, the dilution factor of tube B is now 1 in 100. Mix the tube, transfer 100 microliters again from B to C this time, and the dilution factor of tube C will therefore be 1 in 1,000. Okay, so we're doing 1 in 10 dilutions every single time, and because they're cumulative, that means that it gets 10 times more dilute each time. Okay, and then 1 in 100,000. So these are the dilution factors. Remember, we know how much we've diluted them. We still don't know what the density is, though. Okay? All we know is that tube A is 10 times more dilute than the original. Tube C is 10 times more dilute than tube B, etc. So we do those dilutions, and once we do those dilutions, we are going to inoculate an agar plate with some bacteria or a known volume of these suspensions that we have made. So plate, 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 plate. Right? Okay, and I'll just get rid of these. Right, we are going to transfer a known volume, or not transfer, but we're going to inoculate this with a known volume. Again, let's keep it simple. We'll transfer 100 microliters. It could be anything, but we'll keep it 100. Okay, so we'll call this plate A, plate B, plate C, plate D, plate E. And each of those have 100 microliters transferred onto the plate, spread out using the aseptic technique and so on, and then incubated for 24 hours. We come back after 24 hours, around about 20 degrees C, okay, and we see the following. Plate A, extremely dense still. The plate is completely covered with bacteria, no colonies, okay? Too many individual cells, all still very close together because there's so many of them, and so all the colonies essentially have merged into one, okay? B, we have colonies, but it's still quite dense. And many of these colonies have merged together. Again, not useful to us. We need isolated cells that will form distinct colonies. That's the only way this method is going to be quantitative. Okay, tube C has got a number of colonies, quite a lot, but the cells have been decently spaced out and they're all single colonies. D has got fewer cells, and E maybe has 1 or 2. Okay, so this is kind of the thing that you might expect. It might be that the plate that looks like tube C is your first plate. It might be that the plate that looks like plate C here is your last plate. You never know. Okay, now this is the reason why we did the serial dilution, because this plate and this plate are too dense for us to take any quantitative information from. These last two plates, so let's just make a note of that, too many bacterial cells, too many. That's the problem with these plates. Too many to be useful to us. Again, tube D and E are also not useful to us because they basically have too few bacterial cells, and there's too much error that might be introduced if we start using plates with such low numbers of cells. Okay, so it might not be a very accurate way for us to make an estimation. Okay, because there's just too few cells on these plates. So tube C it's the Goldilocks plate. It's the plate that has got good separation, a good number of colonies to be statistically relevant to us, or statistically reliable, okay? So we are going to use plate C in order to count our colonies and then multiply up by the dilution factor to find out what this question mark is going to be, okay? So how do we do that? So once we have selected plate C, we count the colonies up, and let's just say for example, we have 56 colonies on the plate. 56. So how do we use that number to get back to here and find out what was the bacterial cell number in the original vial, in the original culture? So how we do that is we have 56, and we need to take note of all the things here. Two things. First, what was the volume of the inoculation, right? So 56, first we have 56 colonies in 100 microliters. So if we had 56 colonies, do that here, 56 colonies in 100 microliters, that means there should be 56 times 10 colonies in 1 ml of dilution C. Okay? Make sure that makes sense, right? So 56 colonies multiplied by 10 in 1 ml of dilution C. Because remember, we only took 100 microliters to get 56 colonies. So if we had, theoretically, if we had taken 1 ml of culture C, we would have got 10 times the number of colonies. Or maybe that wouldn't have been a good idea. Okay, so, right, so we got 50, so we got in tube C, we have 560 cells or colony forming units per ml. Now, we know that C, the dilution factor of C was, so if that's 1 in 10, that was 1 in 100, that's 1 in 1,000. So the dilution factor of C was 1 in 1,000. Tube C, we know, is 1,000 times more dilute than the original culture. So we must multiply 560 cells per mil 1,000 times in order to know how concentrated that was, or dense. So 560 multiplied by dilution factor, which is 1,000, okay, gives us 560,000 colony forming units per mil. And that is what that is. Okay, so go over that if you need to. It's the magic of online video, I suppose, right? But make sure that makes sense. And if any part of it doesn't make sense, that is okay. But what you need to do, you can't just say, this doesn't make sense. You have to go back and figure out at what point did it start not making sense. Then you can ask, someone to help you out with that. Okay, so go back and locate the thing that didn't make sense, and maybe leave a question in the comment section or ask a colleague or a teacher. Okay, so that's kind of the basic method. Now, at this point, we should discuss, so why did we need a serial dilution for this? So remember what we said about the Goldilocks plate, the plate that had not too many bacterial cells, not too few, had just the right number for us to count. Okay? Now, the thing is that we don't know which dilution would have produced that kind of plate, right? So if we just did one of the dilutions, it might, so on this day we were lucky, we got plate C and it was right there, but on a different day, if we had a slightly more dense solution or suspension of bacteria, it might be that even plate C might have been too dense. And so on that day, it would have been lucky for us to have the 10 times more dilute plate D and the 10 times even more dilute plate E. Okay, so because we don't know where that Goldilocks, which one of the dilutions is going to produce the Goldilocks plate, that's the reason why we need to make that serial dilution. Okay, so that is that. And the last thing I would say is, remember, this is just a technique to measure this tube, right? So it might be that if you were testing the effect of pH on a bacterial culture, you might have more than just that one tube. You might have some bacteria that you grew at pH 4. If I can write 4. You might have some bacteria that you grew at pH 5, you might have some bacteria that you grew at pH 6, and you might have some bacteria that you grew at pH 7. You would have to do this whole process for each of these tubes to find out the bacterial cell number in each of these tubes to be able to find out the effect of pH on bacterial growth. Okay, now it seems long, and remember, you're not going to actually have to do it. You'll just have to be able to know how to do it and be able to explain it in words. Okay, but yeah, that's all I have to say about that. Remember, I do have a video where you can see me do this practical if you need to see it being done, and this is kind of a supplementary one to go with those, just so that you can understand the theory and the calculation aspect that goes with that practical as well. All right, guys, all the best.